cumulative over basically the whole semester, but in that email through WebCT, I listed the lectures that you need to just review quickly before taking the final. It becomes available on the scheduled day at 6 a.m. May 10th, and is available through 11:55 p.m. May 13th, 2005, in case anybody they're recording this for future years. You have 2 attempts. What I failed to do at the beginning of the semester before posting the syllabus was to readjust the exam 3 so that it clearly did not include the lectures on the urogenital system. You may have noticed on exam 3, even though we had covered that material before that exam, the material didn't show up on that exam. So the urogenital system through all the rest of the semester, the cardiovascular and nervous system and the skeletal system are on the final exam. So I gave the details for that in that email that I had sent out. Also, I had sent messages that we're only covering lectures 1 through 36 for the 35, excuse me, just opened the wrong one, through the semester. So basically the new material on the final exam is lecture 24, the first urogenital lecture, through lecture 35, the last one on the nervous system. Yes?
And you said there'll be 50 questions from 24 to 35 and then 50 questions from 1 to 3?
The first 83 questions cover the new material, lecture 24 to 35, urogenital, cardiovascular, skeletal, and nervous systems, the organogenesis, organ systems development, and then the remaining questions, 84 to 100, are cumulative over all the rest of the material from the semester, but it's obviously not a large number of questions, and they're kind of picked here and there. So I've listed the actual lectures that you should look over to prepare for that. And after taking your first attempt, you'll have a better feel of what to go and look for, I am assuming, so it won't be devastating. It might be a little bit distracting in the first attempt, but take close note to the final set of questions between your first and second attempt. So today we will complete our discussion of the development of the nervous system, discussing the peripheral nervous system, specifically lecture 35. So the peripheral nervous system, we've mentioned it from even very early on, talking about the early establishments of the outgrowing axons for the efferent system, motor systems, both somatic and visceral of the central nervous system, and the sensory neurons that develop from the neural crest cells and all the other supportive cells that are part of the peripheral nervous system. And in essence, today is more of a review, an overview of that information with just a little bit more specifics. There are 12 cranial nerves that develop, and 31, they're all paired, and 31 spinal nerves, so right and left sides. So we do cover the 12 cranial nerves. If you haven't studied those before, that'll be a bit of a new challenge. And then we remind ourselves that ganglia is the term for a collection of nerve cell bodies and their dendrites in the peripheral nervous system. So all neuron cell bodies and dendrites and their axons, wherever they go, in the peripheral nervous system develop from the neural crest cells. And some are, most are sensory, and then others are visceral motor, the postganglionic, sympathetic and parasympathetic neurons. So, for example, the dorsal root ganglion, for the spinal nerves, there's 31 pairs of dorsal root ganglia, the spinal sensory neurons. And in the cranial nerves, there are also ganglia for the sensory functions carried by the cranial nerves, still developing from the neural crest cells and residing in the PNS. There's one exception to that, but I'm not going to get into that level of detail. We had seen the mesencephalic nucleus of cranial nerve 5 in the previous lecture on the development of the central nervous system. That's at least one exception for the sensory neurons of the cranial nerves. But there's another large peripheral ganglion for cranial nerve 5, the trigeminal ganglion, that does fit the characteristic pattern of neurons developing from neural crest cells. The sensory cranial nerve ganglia, as I had stated, are included in this. So there's two sets of sensory ganglia and the autonomic ganglia that are visceral motor. So the developmental characteristics are related to the components developing from the neural crest cells, as we said. So there are somatic sensory neurons. There are visceral sensory neurons. And we'd even discussed a little bit in the previous lecture the general somatic, general visceral, and special visceral. And we'll get into a bit more detail of that today. But for now, we're just seeing that on the sensory component, there's both sensory and visceral. There are satellite cells. These I've mentioned all along. So neural crest cells develop into these supportive cells surrounding the neurons within the ganglia. They're called satellite cells. Along the extensions of the fibers, both the sensory and motor fibers, these neural crest cells will differentiate into Schwann cells, either protecting the unmyelinated fibers or myelinating the myelinated fibers. The adrenal medulla is another target of neural crest cells. Adrenal medulla basically represents postganglionic sympathetic neurons, but they just don't differentiate into neurons. They reside in the center of the adrenal gland and stay balled up, basically. They just don't take on the morphology of neurons, but basically they do the same thing as a postganglionic sympathetic neuron that you'd find someplace else, synthesizing and releasing norepinephrine, small amounts of norepinephrine, but predominantly epinephrine or adrenaline, right? Let's not get that confused. And melanocytes within the skin, and then chromaffin cells. These are scattered cells similar to the adrenal medulla, found in different areas of the body, such as in the gut tube, the enterochromaffin cells that synthesize and secrete serotonin. This slide is showing us two important aspects of neural crest cells. There's a top set of panels and then a lower panel. The top set of panels on the right-hand side, you will recognize this from the lecture on the spinal cord, where we had mentioned that this is a scanning electron micrograph of a mouse embryo cut in cross-section and scanned or viewed from the cut end, whether it's rostral view or caudal view, doesn't matter. And we identify the neural tube, and we see the neuroepithelial cells within the neural tube, and of course we also recognize the notochord. And then at the junction of the dorsal plate or roof of the neural tube, the site of closure of the neural crests, the margins of the neural folds, is the junction with the overlying now surface ectoderm continuous across the surface. And at that junction, there was a set of cells, the neural crest cells, that migrated away. They were somehow triggered to differentiate down a different lineage, still neuronal and supportive, or glial if you want to think of it that way, distinct from those that remained within the neural tube. They take on a migratory function, so they actually are motile. They do move to new locations. And then here on the right-hand side, they're the rightmost panel, they are artificially colored in red so that we can see them in their passage ventrolateral away from the dorsalmost portion of the neural tube. In the upper left-hand panel, we see this illustrated with colored identification of the different cell types. We see the neural plate cells, these tall columnar cells in the pale green. And we see it's a simple columnar epithelium. Ultimately, we know that becomes pseudostratified and thickened, and then finally multiple layers forming the marginal zone, the intermediate zone, and the ventricular zone. But at this stage, it's just simple columnar epithelium, these tall columnar cells forming the neural plate. At the margin of the neural plate, where the neural fold would be, there is an intercellular connection with the surface ectoderm cells. And just at that junction, some of the neural plate cells, or they're actually neural crest cells, but they're more closely related to the neural plate cells than to the surface ectoderm cells. They take on this different lineage of development, become migratory, and begin to move ventrolateral to this site of origin to take up residence at the future location of the dorsal root ganglion. So not all of them migrate to great distances, but some of them will. Well, how will those cells, neural crest cells, migrate and differentiate? What are the factors that would be involved? Well, let's first consider the stimuli for the further differentiation of the adrenal medulla cells versus the postganglionic sympathetic neurons. So these do migrate quite great distances. Might not seem like much just to go from the location where the primordia of the adrenal gland would be to migrate on over and take up residence in the mesenchyme and further differentiate into the adrenal medulla. And then all the locations where there would be sympathetic ganglia, we would see this upper panel of cells migrating to those areas. So it starts when things are relatively close, so signaling is a more successful probability. If you had to have a cell migrate from the edge of your brain to the tip of your finger as an adult, obviously that would be much more challenging than when the finger or primordial cell is somewhat adjacent to that area. I don't think any cells migrate from that to the fingertip, but anyway, it makes the point for us. So let's first think about differentiation, and then along that line of thinking of differentiation, we'll look at events related to the motility of these cells. All of this, as we know, this differentiation as well as going through a motile stage is very much dependent upon differential gene expression. At one point in time, there will be exposure of the cell to chemicals in the interstitial fluid compartment, the extracellular fluid compartment, and there will be some type of receptor-mediated signaling that will impact gene expression and thereby impact the morphology, structure, and function of the cell. Same thing for telling the cell to express proteins to interact with the extracellular matrix, to walk along the connective tissue fibers and find your final site of residence. So here we're just thinking of differentiation along two lineages that are predominantly morphological and not really chemical differentiation as far as the secretory substances. So we see first all the way on the left, the pluripotent neural crest cell will be migratory and eventually, some gene expression just changes over time. We don't really understand the full regulatory, the full spectrum of the regulatory mechanisms. There just be, once one event begins, then there's a progression that might only be an internal signaling system to have a different gene turned on a little bit time later, a different gene turned on a little bit time later. So there are some cases where that occurs, at least as far as we know now, independent of any extracellular signal. But the initiation of the event might have been triggered by a very early extracellular signal, and then once it started internally, then it regulates itself for the sequential expression of developmentally important genes. So this very likely is what is going on, to have the neural crest cell take on this lineage to become a bipotent precursor cell. So we remember totipotent, multipotent, pluripotent. Now we're down to bipotent. So you can use any prefix on this, tetrapotent, tripotent, whatever it would be, ultimately down to bipotent, and then ultimately and finally as unipotent. This cell will only develop into sympathetic neurons if its precursor, this bipotent cell, was in a location where it was bathed with this, I think it's a fibroblast growth factor 2, the FGF2, and then it becomes an NGF nerve growth factor competent cell. So this extracellular signal tells this cell to express genes for the receptors of nerve growth factor, such that when this cell ends up in its final location and the local nerve growth factor is present, it'll trigger, by binding to the receptors that are now expressed by this cell, trigger the termination of migration and the final differentiation of the cell morphologically and, in this case, neurochemically, as a secretory neuron that's going to synthesize and secrete predominantly norepinephrine. So there's one gene with the corresponding enzyme that's different between a cell that will synthesize norepinephrine versus one that will have this final enzyme, phenylethanolamine and methyltransferase, no big deal for us here, that'll convert the norepinephrine into the epinephrine. So there's a slight difference in the gene expression of this lineage to produce the postganglionic sympathetic neuron that will synthesize and secrete norepinephrine versus the one that will follow from the bipotent stage with exposure to glucocorticoids. So these cells will somehow have found their way to the site where the adrenal cortical cell precursors are, and when those adrenal cortical cell precursor cells begin to synthesize and release the corticosteroids, the glucocorticoids, those bipotent precursor cells that are there and exposed to the significant levels of the glucocorticoids will have their genes targeted such that the cell will become non-migratory, will begin to express the genes for the synthesis, the enzymes that synthesize not only norepinephrine, but that one final one, the phenylethanolamine and methyltransferase, that will convert the norepinephrine to epinephrine. And under continued glucocorticoid stimulation, this chromaffin precursor cell will reach terminal differentiation as a chromaffin cell. And this is a chromaffin cell specifically located in the adrenal medulla. There are other peptide molecules that are genes that are turned on and expressed, and the peptides released by these cells versus these cells. So it's much more sophisticated than what I've mentioned, just focusing on whether it synthesizes norepinephrine ultimately or epinephrine ultimately. But these remind us that differentiation requires differential gene expression most often that is dependent upon an extracellular signaling system. The neural crest cells are illustrated again in this set of figures. Their site of origin dorsolateral to the neural tube. Some remain, not all of them move away, and some will migrate on out to different target locations. As well, these arrows are representing the extension of the sensory fiber from the neural crest cells that will remain as in the dorsoroot ganglia. So we not only have migratory neurons, but migrations of the extensions of the sensory fiber going on out, and the motor fibers that will have started coming out of the ventral spinal cord will proceed with that as well. But let's now just focus on the cellular migration. So this other panel shows us the migration of the neural crest cells, and the graph is over time on the y-axis, and relative amounts of different molecules either in the extracellular matrix or expressed by the neural crest cells. So initially they are located in the neural folds, then they migrate through the tissue, and then they will, it's arrival, not very easy to read. There will be the arrival at the site of differentiation, as we had emphasized in the previous slide. So something happens between their original location and their target location between the surface proteins on these cells and the extracellular matrix. So there are changes in the cell surface and the extracellular matrix. So this NCAM is neuronal cell adhesion molecule. So in the pre-migratory phase, the expression of this NCAM is high in the neural crest cells. So it's expressing these adhesion molecules on its surface. So it's attached to the extracellular matrix, and it's not going to go anywhere. Somehow that gene expression is turned off at the transition to the migratory phase. And we see in the extracellular matrix that fibronectin and laminectin levels increase, so the mesenchymal cells turn on their genes to increase the expression of that, and with the low levels of NCAM and the high levels of fibronectin and laminectin, these neural crest cells now can attach and pull itself up, kind of the snail, the podocyte motility that we're familiar with, such as the amoeba cell that we studied very early on. Once reaching the final site of differentiation, the reverse happens. We reduce, the mesenchymal cells are no longer producing the high levels of fibronectin and laminectin, and these cells begin to express their gene for their adhesion molecules and stick in place. So this portion is a review. Most of the neurons start as the bipolar cells and become the pseudo-unipolar that we had seen before. The exception are the bipolar neurons associated with cranial nerve 8. The peripheral nerves are enveloped by the connective tissue, a mesodermal derivative. So I had stated before that the Schwann cells and the satellite cells come from neural crest cells, but the connective tissue and the vasculature are of mesenchymal origin. So this is a figure that we had seen before. The rounded neural crest cell will begin to differentiate into a bipolar type of a neuron, a neuroblast. Those two processes migrate together on the surface of the cell and become fused as one. Appears as unipolar, but that splits, so it's called pseudo-unipolar in the adult case. The spinal nerves will develop with the motor nerves from the spinal cord around week 4, beginning to extend out of the ventrolateral portion of the neural tube. And those efferent axons will join the extensions of the sensory neurons that are the neural crest cells and the dorsoroot ganglion. The cell bodies for the motor efferents will originate from the basal plate of the spinal cord, as we had said. The sensory neurons originating from the neural crest cells will be in the dorsoroot ganglia. These two sets of fibers, the motor and the sensory from the dorsoroot ganglia, will join together, as we had discussed in the formation of the vertebral column, so that they are joined together as a mixed neuron, spinal neuron, having both sensory and motor components in between the vertebra. So the motor root combines, forms the mixed spinal nerve. Then the mixed spinal nerve will divide There'll be a dorsal branch and a ventral branch, and then they target their respective areas, and any additional detail to that is beyond the scope of this course. But it emphasizes the point that these motor and sensory fibers somehow find their targets early and follow them where they're going to go and stay there and maintain that innervation. The final point of discussion then are the cranial nerves. There are 12 cranial nerves. There are those that have somatic efferents. The cells are derived from the basal plate of the the forebrain, of the rostral levels of the neural tube. So, for example, cranial nerve 12, the hypoglossal, has general somatic efferent to the muscles of the tongue, and general somatic afferent, so we know where our tongue is located. Cranial nerve 6, the abducens, has general somatic efferent and general somatic afferent. They innervate the muscles, the lateral rectus muscles, some of the extraocular muscles, so that we have both the motor to move it and the sensory to tell when it's stretched and all that stuff, the sensory information from muscles. Cranial nerve 4, the trochlear, also has general somatic efferent and afferent, and this is to another extraocular muscle. And cranial nerve 3, the oculomotor, in addition to the GSE and GSA, have the general visceral efferent. There is a parasympathetic component of this. Cranial nerves 3, 7, 9, and 10 include preganglionic parasympathetic components. So the targets of all these are not a responsibility for you in the class. It's more of understanding the components within each of the cranial nerves. The nerves of the pharyngeal arches, for example, the trigeminal, we stated, has special visceral efferent, special and visceral because it has to do with that special function of the rostral end of organisms, as we had said, but even more so because it innervates muscles that are generated from mesenchyme that came from neural crest cells, that characteristic that is unique to the pharyngeal arches. And this was the cranial nerve we said was found in the first arch. The second arch has cranial nerve 7, so it has special visceral efferent, special visceral afferent for taste, and general visceral efferent 3, 7, 9, and 10 contain the preganglionic parasympathetic fibers. Cranial nerve 9 was in the third arch. That's the glossopharyngeal nerve. It has special visceral afferent for taste and general visceral efferent 3, 7, 9, and 10. So this is the parasympathetics within cranial nerve 9. The cranial nerve 10 has special visceral efferent, still some taste fibers, special visceral efferent to the musculature, special visceral afferent taste, general visceral afferent for the sensory innervation of the tongue and the pharynx, and also the esophagus, and then general and all the rest of the midgut, as we know, for the intestine. Then general visceral efferent for its parasympathetic component, 3, 7, 9, and 10. Again, then cranial nerve 11, the spinal accessory, has special visceral efferent. It is only innervating muscles that developed from that specialized mesenchyme in the pharyngeal arch area. We didn't describe, well, we did have cranial nerve 10 or 12 earlier on. So the special sensory ones are obviously the olfactory, cranial nerve 1, special visceral afferent. So it's a special sense. It's visceral because everything is dissolved in the mucus. The cranial nerve 2, the optic, special somatic afferent, somatic because of the physical light. And cranial nerve 8, the vestibular cochlear, special somatic afferent, the otoliths and the hair cells and all that stuff, making it seem as a somatic type of a receptor function and not really a visceral one. So you'll have to match those types of functions with the cranial nerves, recognizing their Roman numeral, and as well matching the cranial nerves to their Roman numeral themselves. This slide is in here for a bonus question. You will not, this is irrelevant to what we will be doing. It has not worked into the electronic format of the exam. So this completes our discussion of the development of the human organism. I appreciate your time and attention to the semester, and I hope you all have enjoyed the WebCT aspect of this course. Thank you.